Hey everyone, this is Professor Ways, and today we're going to do a review of the eye. So a lot of what we're going to cover is terminology, and most of this is going to be review from anatomy over the summer, with a few new things added to match what the instructional objectives are for this section of your Clin Med course. So just to orient ourselves before we get started, for a lot of these terms, I'm going to have things in gray, and gray are the things you should pay most attention to as usual. And sometimes you'll see these parentheses with a medical terminology format of a word. And what this means is, for example, the one I just circled, if I'm talking about the iris, then anytime you see something that says irido and then a suffix after it, then it has something to do with the iris. For example, if you have an iridotomy, that would be a specific medical procedure that is basically putting a hole in the iris to relieve pressure from glaucoma. So we begin with structures of the external eye, beginning with the eyelids here, also known as palpebrae, and then the space in between them, marked by these arrows here, is known as the palpebral fissure. And when our eyes are open, the fissure is large. When our eyes are closed, the fissure is just nothing more than a slit. On the lateral aspect of the eye, we have the outer or lateral canthus. And on the medial aspect of the eye, we have the medial or inner canthus. And you can tell the outer canthus from the inner canthus, or lateral from medial canthus, because the medial canthus is going to have a little more of a fleshy appearance that's going to contain the puncta, which are the drainage sites for tears as part of your nasolacrimal duct or your tear duct, as well as this part here, which we'll go over a little bit later, the lacrimal lake. Looking at the eyeball itself, the white part of the eye that we can see is known as the sclera, and the colored portion is known as the iris. And the iris, of course, is made up of muscle, and when that muscle contracts or relaxes, that will then change the size of the pupil, allowing more or less light into the back of the eye. And then finally, we have the limbus or limbic margin, which is not directly visible here, but you can see it as the border between the iris and the sclera. But what it really is the border between the cornea, which covers the iris, and the sclera. So again, the limbic margin or the limbus looks like it's the border between the pupil and the sclera, but really it's the border between the cornea and the sclera. And the pupil just happens to be, the iris just happens to be sitting in the same location. All right, continuing with the external eye, this here in red would be the eyelid or the palpebra, upper palpebra or superior palpebra. And that will raise when the levator palpebrae superioris muscle contracts. So that muscle is right here, and when that contracts, it'll pull the eyelid up, thus opening your eye. Remember from the previous slide that the white part of your eye is known as the sclera, and then the front part of your eye right here that joins with the sclera and forms what we referred to last slide as the limbic margin. This would be the cornea, right? And if you look over here, cornea is generally referred to as kerato. So if we have something called a keratoplasty, that would be a surgery on the cornea, for example, a corneal transplantation. So again, important to know it's the cornea, but also important to know that kerato refers to a procedure or something to do with the cornea. Now, covering the sclera is a very thin vascular membrane that not only covers the sclera, but is continuous up into the palpebra and down on the posterior side of the palpebra, and that's known as the conjunctiva or conjunctiva. All right. Of course, you see the med term form here, so conjunctivitis would be inflammation of the conjunctiva, for example. And then where the conjunctiva that's covering the sclera joins with the conjunctiva that's on the surface of the palpebra, we call the superior conjunctival fornix, or just fornix of the eye, if you will. And fornix simply means arch, so you see this is an arch of the conjunctiva. And then, of course, within the eyelid, we have the tarsus, which is a connective tissue plate within the eyelid, and the tarsus contains the tarsal glands. And over here you see a description of what the tarsal glands actually do. So I'm not going to go over it, I'll let you guys read that. Finally, going back to the cornea right here, it's important to understand that the cornea has 5 layers to it from the outside going in. So the outermost layer is a layer of epithelial tissue, and you can see it's stratified in nature, so it's that makes it protective layer of cells. Underneath that, in fact, underneath the basement membrane of the epithelium, we have what's called Bowman's layer. And Bowman's layer is a strong collagen layer, so again, another added layer of protection. It makes that cornea kind of tough. Then the next layer is, of course, the stroma, and the stroma is made up of collagen as well, but it also has a lot of water in it, making it a very pliable structure. Underneath that, we have the Descemet's membrane. And again, that looks very similar to what you see with the Bowman's layer. And it's also made out of collagen, but a different type of collagen. It's actually tougher collagen than what you see in the layers above it, again, for another added layer of protection. And then just below that, we have the endothelium. And this is very similar to the endothelium that you will see in blood vessels. Remember, it's that smooth, shiny surface that is between the actual blood and the layers underneath the endothelium. And what it does in the eye, notice that it's only like a layer thick of cells, is that it makes the collagen that you'll find in Descemet's membrane, but it also removes excess water from the stroma so that the stroma doesn't become clouded, and therefore affect the way light enters the eye and thus affecting vision. All right, so continuing with the external eye, we have the lacrimal apparatus, and remember the lacrimal apparatus is on the medial portion of the eye, associated with the medial canthus. And anytime we see this term DACR, that's going to refer to anything to do with that lacrimal apparatus. So again, for example, if we have the term dacryoadenitis, that's referring to inflammation of the lacrimal gland. The term DACR refers to that lacrimal apparatus. All right, as part of our lacrimal apparatus, we have the lacrimal lake, which is this fleshy portion right here where the fluid that is in the eye will collect. And of course, remember the lacrimal gland is over on the lateral portion of the eye. Ducts allow fluid to drain into the eye, and then it wipes towards the lacrimal apparatus. So that lacrimal lake will collect that. And then by capillary action, fluid will move into the puncta and into the lacrimal canaliculi, and then collect within the lacrimal sac and drain into the nasolacrimal duct. So if you have an excess of tears that are draining into this lacrimal apparatus in the medial canthus of the eye, then you'll tend to get a runny nose because all of those tears are draining into the nasal cavity, and thus making nasal drainage as well. All right, so now we'll start moving into more of the internal eye. We'll still talk about some external structures, but we're going to relate those external structures to the internal structure of the eye. So here again we see the cornea, and we just talked about the cornea and what it is and the 5 layers that are associated with it from outside to in. The cornea is just anterior to what we call the anterior segment of the eye. An anterior segment is divided from the posterior segment by this lens right here. So the anterior segment is divided into, just behind the cornea, the anterior chamber right here, and then the posterior chamber right here. And this purple structure on either side right here, I'll do that in green, that would be your iris. All right, so the anterior chamber and the posterior chamber are separated from each other by the iris, although they are connected through this opening within the iris called the pupil. And then within the anterior segment, you're going to have aqueous humor, which is a liquid. And we'll talk about how that's formed and what it's for and how it leaves the eye in a couple slides later on. All right. At the posterior border of the anterior segment, we have the lens. And again, anytime we're talking about the lens, if you see phaco something, that is referring to the lens. For example, if you hear the term phacoemulsification, that is a form of cataract surgery. You'll also see the lens, or the combining form of the lens, referred to as phaco with a C instead of a K. All right, associated with the lens, we'll see here the ciliary body. Actually, the ciliary body is right here. And this is a 2D structure, so imagine it's going all the way around the eye, all the way around the lens. And anytime you see the prefix cycle, that's referring to the ciliary body, or a disease or a surgery of the ciliary body. And of course, the ciliary body is a muscular structure that's responsible for putting tension on the suspensory ligaments attached to the lens, and that will change the shape of the lens, and we'll show this in a couple slides, thus allowing your eye to accommodate to near and far vision. And you've done the accommodation test in your physical exam lab, so this is the physical basis of what's happening with the change in shape of the lens to facilitate this accommodation. All right, extending from the anterior eye backwards, we have the sclera. Remember, that's the white part of the eye, and it is a dense, thick, protective covering going all the way to the back of the eye. Just underneath that, we see the choroid layer, and the choroid layer is another protective layer of the eye. And just inside of that, and it's usually pigmented, and just inside of that we see the retinal layer. And the retina, which we'll talk about in more detail later on, is basically the nervous system component of the eye, which detects light or changes in light and transmits them to the optic nerve to go into the central nervous system to be interpreted. All right, where all the retina converges here to go into the optic nerve, that is referred to as the optic disc, and the optic disc prefix is papille. So if you see something like papilledema, that would be a swelling of the optic disc. Near to the optic disc, we'll see something called the fovea centralis within the macula lutea. And again, we'll talk about this a little bit later on. And remember, that is the most, where the clearest point of vision is located, where you're going to have a high concentration of color receptors, the cones, to give you clarity of vision. And finally, this section of the eye right here, which contains the retina, that is referred to as the posterior segment of the eye, and the hollow space that was just circled is referred to as the the vitreous chamber. The vitreous chamber, which contains vitreous humor. And now vitreous humor is a little bit different than aqueous humor. If you remember, aqueous humor is liquid and it's constantly moving, whereas the vitreous humor, if you've ever done a dissection of an eyeball, like cow eyeball, maybe in undergrad or high school, the vitreous humor is like jelly. It's a little bit thicker, and part of the point of the vitreous humor is to put pressure on the external wall of the eyeball to, one, maintain pressure, and to, two, keep that very loose fitting retina tight against the wall of the eye, so that you don't have waves or ripples within the retina that would affect the way light is being interpreted by the eye. One more thing, if you notice in the bottom left corner right here, I have a frisbee or a disc. And the reason why I put that in here is I want you to think of the front of the frisbee right here as the front of the eye, where you see the cornea right here, and then underneath would be where the anterior chamber is located. And this ridge that's going all the way around here is basically these angles that you're seeing here between the cornea and the iris right here. So I don't want you to get the feeling that you just have two angles right here. It is actually a circle going all the way around. So when we talk about the angle, we're talking about the circular angle all the way around, not just two distinct parts of the eye. All right, a little more detail about some of the structures, starting with the iris right here. Remember, the iris, which is the colored part of your eye that surrounds the pupil, which is the part of the eye that allows light to go in, is made up of muscle. Now, there's two types of muscles within the iris. You have the dilator muscles, and you notice that these dilator muscles are arranged in a perpendicular fashion all the way around the pupil. And so when they contract, they're going to pull things outward, and that's going to result in dilation of the eye. That's why they call it the dilator. And then in the center, you have longitudinal muscles running all the way around the pupil. And when those contract, of course, they are going to make the pupil smaller. So they're responsible for constriction of the pupil. So you have your dilator and your constrictor. When the constrictor muscles relax, you get a dilation to normal size. When the dilator muscles relax, you get a constriction back to normal pupil size. Okay, so if you remember from your physical exam, normal pupil size in adequate light is about 2 to 4 millimeters. And then when you go into like a dark room, it will dilate to let more light in to about 4 to 8 millimeters. And if you go into a really bright room or sunlight, you'll have a constriction down to about 1 to 2 millimeters. Now, if we have a pupil that is excessively constricted, so it's like really small for the amount of light that's in the room, we refer to that as miosis. Now notice that the pronunciation of miosis is very similar to the germ cell division, meiosis, but the difference is in the spelling. Meiosis, as it refers to the eye, is spelled M-I-O-S-I-S, whereas meiosis, referring to cell division or the type of cell division, is M-E-I-O-S-I-S. All right, so make sure that you can tell the difference between those two. If the pupil is dilated, we refer to that as mydriasis. All right, pupillary dilation. And then in addition to pupils being about 2 to 3 millimeters under normal conditions, they should also be equal to each other. All right, if the pupils are not equal to each other, we refer to that as anisocoria. All right, uneven pupil dilation and constriction. So I encourage you to go to Google type in anisocoria and look at the images, and you can get a good example of what some of these, of what that would look like. Next, we're going to move back to the posterior chamber of the anterior segment of the eye and focus on these ciliary bodies right here. So you notice that in the ciliary bodies we have this ciliary muscle, and then we have these ciliary processes, and the ciliary processes are attached to these suspensory ligaments, and the suspensory ligaments then are in turn attached to the lens. So you can imagine if we pull on the suspensory ligaments, that's going to change the shape of the lens from something that looks like a circle to something that's more oblong. And then if we relax those fibers, or those suspensory ligaments, then we return to something that is more of a round shape. All right, so if we're changing the shape of the lens, then we're going to be changing the shape, or changing the way that light will enter the back of the eye. And again, that's referring to the process of accommodation that allows you to adjust from near vision to far vision pretty seamlessly. So if we go over here to the left and look at the ciliary body and the lens and the suspensory ligaments, also known as zonular fibers, all right, from the anterior view instead of the side view, which is to the right, we see that the ciliary muscles on the left side of the picture right here, they are relaxed. And when the ciliary muscles are relaxed, what I want you to notice is that these zonular fibers or suspensory ligaments are tight. All right, so when the ciliary muscles are tight, or are relaxed, the suspensory ligaments are tight, and your lens is more of an oblong shape. But when those ciliary muscles contract, all right, and remember, when muscles contract, they shorten, and when muscles shorten, they look a little fatter, right? So that's why you see this difference in what the muscle looks like here, because they're contracted, so it's a little bit fatter. But when those muscles contract and get larger and take up more space, then that's going to allow these suspensory ligaments to go slack. And when they go slack, of course, you're not putting tension on the lens, and the lens returns to more of a round shape. So what this does is it allows your lens to adjust to what you're looking at and basically focus appropriately. So if you close your eyes and then you open them looking off into the distance, what's going to happen is your ciliary muscles are going to relax, making your lens thinner, right, stretched out a little bit more, and that is going to allow the light coming from those distant sources to focus appropriately on the fovea centralis within the retina of the eye. And then if all of a sudden you drop something right in front of your face, let's say about 12 inches from your face, well then your lens is going to have to adjust to that near vision now. So these ciliary muscles are going to contract, allowing those fibers to go slack, and your lens is going to get fat, which is going to change the way the light bends through the lens, and thus allow the light to once again focus on the fovea centralis, making the vision clear again. Right, and again, that process, so this would be associated with this picture, that process is called accommodation, near-far accommodation. Please refer to the video in your PowerPoint presentation or visit the YouTube site associated with that video for more information on how ciliary action works. Okay, so moving on to the retina. And remember, the retina is that layer that is being held in place by the vitreous humor, and it is where light is detected and sends signals to the optic nerve that enters the central nervous system. And there are 10 layers to the retina. So if you think of this area right here as being the vitreous humor, all right, and this down here is the choroid, then all of the layers in between are the layers of the retina. So not that you have to specifically know all the layers of the retina, but be familiar that there are 10 layers, and each of the layers has a slightly different features and does slightly different things. So we have the internal limiting membrane, which is kind of the barrier of the retina, the outer barrier or internal barrier, if you will, facing the inside of the eye. Then we have the retinal nerve fiber layer, so the nerve fibers that are going out and then converging onto the optic nerve. That's what this retinal nerve fiber layer is. Then we have the ganglion cell layer, the inner plexiform layer, and you notice that there are a lot of connections being made with the inner plexiform layer, the inner nuclear layer, which contains several different types of intermediary cells, the outer plexiform layer, which again, plexiform you should associate with connections being made, the outer nuclear layer, and by outer nuclear layer we are referring to the nuclei of the rods and the cones, the external limiting membrane, so really where the external border of the retina would be ending, and then the photoreceptor rod and cone cell inner and outer segments, so this would be the spot that's actually detecting light. And then we have the pigmented epithelium, and again that is associated with the choroid and makes sure that the section underneath the rods and the cones is darker so that when light hits it, it doesn't scatter and cause blurry vision. So one of the importance of mentioning these membranes here, since we're not holding you responsible for knowing all the specific layers and exactly what they do, is that light is coming through in this direction. So light has to go through all of these layers of the retina in order to hit the rods and the cones, and then the electrical signals generated go back up this way and then into the nerve fibers that end up with the optic nerve. So despite the fact that you're going through all these layers and there's a lot of cells there, vision's not compromised because if you recall, cells don't really have a color unless you're melanocytes or pigmented cells. So light traverses through them pretty readily, and the distance between the internal limiting membrane and the photoreceptor cell inner and outer segments is very tiny. Even though it's blown up here and it looks like a lot of space, it is less. It is a very, very, very short space. In fact, the thickness of the retina is about 250 micrometers, right? Micrometers, so that's like a quarter of a millimeter in thickness. So light is going to pass through it very readily, and these layers are not going to interfere with how light is detected. All right, within your eye, you have general terminology that refers to more specific structures. These are called the uveal tracts, and you have the anterior uveal tract, which is made up of the iris and the ciliary body. So these right here, that would be the anterior uveal tract, and then that forms a continuity with the posterior uveal tract, which is the choroid. All right, so the choroid is continuous with the ciliary body and the iris. All of those together make up the total of the uveal tract. And the reason why we want to mention this and why it's important is that if you get a condition called uveitis, that would be inflammation of your uveal tract. All right, so knowing that you know where the inflammation is going to be located, what structures it's associated with, and how you can best go about trying to alleviate that, or even more so, how it's going to affect the eye. All right, going back to physical exam, your fundoscopic examination. Remember, your fundoscope is looking at the fundus of the eye, which would be the retina, the back portion of the eye. And remember, you have two sides when you're looking at an eye. You have the temporal side, which is towards the temporal part or the lateral part of the eye. And then you also have the nasal side, which would be the medial portion. So, not labeled here. Remember that the macula and the fovea are lateral or temporal to the optic disc. All right, so the macula, the fovea centralis would be that spot of highest visual acuity, and the macula would be the area that's just surrounding that. And then the optic disc, of course, would be where your where your optic nerve exits the eye. And of course, since that is a bundle of nerve fibers coming from the retina, and there's no actual retina in that area, that is also going to be considered a blind spot of your eye. And you also notice that you have blood vessels coming out of that, arteries and veins, that spot as well. So you're not going to be able to see out of it because of that, because of the way that it's structured. Now, remember that the optic disc is this wider portion, and the optic cup is a smaller portion within the optic disc, and the cup is about 40% of the size of the optic disc. Now remember, you also use the optic disc as a measurement of size or distance within the retina. So you could say that the macula is X number of optic discs away from the optic disc, for example, or a bulging artery would be X number of optic discs away from the macula, or X number of optic discs away from the optic disc. All right, speaking of arteries and veins, you should be able to tell arteries from veins. If you look at this picture right here, the arteries are a little bit smaller, tighter. That's because of the way that arteries are, the wall is structured. Remember that. Veins tend to be a little more floppy and hold more blood, therefore they appear bigger. So you can tell the difference between arteries and veins, and the artery to vein ratio is about 2 to 3. So you have about two arteries for every 3 veins. And that's what's visible to you. So you're kind of looking at that to get an appreciation for the general health of the vascularity of the eye, or the retina rather. All right, we mentioned earlier that the anterior and posterior chamber of the eye, those chambers that are just anterior to the lens, part of the anterior compartment, are filled with aqueous humor. And remember, that's the liquid that is constantly circulating within the anterior segment of the eye. Now, aqueous humor is made within these ciliary bodies. All right, the same ciliary bodies that contain the ciliary muscles that apply tension or relaxation on the suspensory ligaments attached to the lens, they produce the aqueous humor. And the aqueous humor flows through the posterior chamber of the eye, through the pupil, and into the anterior chamber of the eye, filling it up. Now, you don't want fluid just staying there because it's going to become stagnant and can cause problems. So within the excuse me, within the angle between the iris and the cornea, we have this almost like a sieve network called the trabecular meshwork. And the trabecular meshwork's function is to drain the aqueous humor, that drainage enters into this structure called the canal of Schlemm, and from the canal of Schlemm the fluid re-enters venous circulation and then gets returned to the body and to the rest of the blood. So in this slide you see the aqueous humor entering the trabecular meshwork right here, the canal of Schlemm, and the canal of Schlemm is connected to these small veins that exit the eye and become continuous with veins returning to the heart from the head. So we'll show this video that will hopefully help you understand how the flow of aqueous humor goes. And for this video as well, please refer to the video that's within your PowerPoint presentation or the YouTube that it's attached to for how aqueous humor flows. Now we want to look at our extraocular muscles. Remember, the extraocular muscles are those which are in gray right here, your superior oblique, your inferior oblique, and then your rectus muscles, superior, inferior, medial, and lateral. Now the rectus muscles, they will move the eye in the direction that the muscle is located. So the lateral rectus will move the eye lateral, the medial rectus will move the eye medial. The oblique muscles, on the other hand, will rotate the eyeball, and since the eyeball is located at an angle compared to the socket, it's actually going to end up making the eyeball look up or look down when the eye is oriented medially. All right, also to remind you that all of the extraocular muscles are supplied by cranial nerves, and all but two, including the muscle that surrounds the pupil, the iris, as well as the levator palpebrae superioris, are supplied by cranial nerve 3, which is the oculomotor. Cranial nerve 4, which is the trochlear nerve, supplies the superior oblique muscle, which is going through a trochlea or pulley to be able to work properly. That's supplied by cranial nerve 4. And cranial nerve 6 supplies the lateral rectus muscle. So that would be this one right here. All right, now go back to your physical exam and remember that your eyeball is supposed to be pointed outward at an angle compared to the front, but we look forward. So that means when we test our extraocular muscles, we want to put them in a position where those muscles are going to be isolated. So that is our 6 cardinal fields of gaze. Looking upward and lateral tests the superior rectus. The inferior rectus is downward and lateral. Lateral rectus is just lateral. Medial rectus is just medial. Inferior oblique is looking upward and medial. And superior oblique is looking downward and medial. Now you test both eyes at the same time and you get all of these fields of gaze at once, but just remember that as you're testing the superior rectus muscle in one eye, you're testing the inferior oblique in the other eye, because both eyes are going to move together. If they don't move together, that means you have a disconnect between your cranial nerves and you've got other problems going on. So once the visual field in front has been projected onto the retina, there's a certain pathway that the visual information takes to the optic centers of the brain. So just as a reminder what these visual pathways are and how they work, the lateral portion of the left visual field will enter the medial portion of the left eye, and the medial portion of the left visual field will enter the lateral portion of the right eye. And then, of course, the lateral portion of our right visual field will enter the medial portion of our right eye, and the medial portion of our right visual field will enter the lateral portion of our left eye. And from there, the wiring is pretty intuitive. So the lateral portion of the left eye will travel down the optic nerve, bypass the optic chiasma, and travel to the lateral geniculate nucleus of the left side of the brain. And the medial portion of the right eye will travel down the optic nerve of the right side, cross at the optic chiasma, and synapse onto the lateral geniculate nucleus on the left side of the brain. All right, then the same will happen for the other side. The medial portion of the left eye will cross over into the right side to the lateral geniculate nucleus of the thalamus, and the lateral portion of the right eye will bypass the optic chiasma and stay on the right side, entering into the lateral geniculate nucleus on the right side. And from there, both pathways will head into the visual cortex of the occipital lobe to be processed. So to summarize that, if we look at this part right here, the left hemisphere of each eye transmits to the left hemisphere of the visual cortex. So two eyes on opposite sides processing left hemisphere vision are going to go to the same side of the visual cortex, whether that be on the left or the right. So you want to keep those pathways in mind. All right, again, they're going to synapse in the lateral geniculate nucleus and then go to the primary visual cortex where that image is going to be reconstructed and interpreted. All right, so when visual deficits happen, the observations that the patient is going to make are going to depend on which part of the visual field is affected. And although we haven't necessarily talked about this stuff, we will start with glaucoma. So imagine if you have glaucoma and the periphery of your eyeball, so around this area right here, all the way around is affected. You will see basically just in the center of your field of vision. So you can mimic this by just putting your fist up to your eye and looking through a small hole. That is basically what you would see with glaucoma. Do that in both eyes and kind of simulate for yourself what that might look like. Now, if we have a central scotomata, then we have some sort of problem right in the center of our eye. Okay, and you might talk about these disorders when you get to that particular section of Clin Med. So I'm not going to go into, what these are or how they happen, but what you would see is basically no blind spots in the center of your field of vision. So take a finger and put it in the center of your eye and see how much of one eye, close the other eye, and see how much you can see of your vision. And that's what that number 2 there would look like. Now, when we're talking about deficits in the nerve pathway, we will outline them according to the number and what happens at each number. So number 3 would be if you have a damage to one. Typically it would be one, it could be both depending on the disease and or the event that occurred, your optic nerve. So if you have a loss in your optic nerve at number 3, then that means all visual signals coming from the right eye are going to be cut off. And so you're only going to be able to see with your left eye. All right, that's pretty straightforward. If we have a disorder which affects the optic chiasma, only the optic chiasma, the part where the nerves are crossing each other. Notice that the lateral parts are unaffected, just the medial parts that are crossing over. So if that's the case, then only the lateral portion from the left eye and the lateral portion from the right eye going to the lateral field of vision from the right eye, going to the medial portions of each eye will be affected. So that means you're only going to see the medial portions in each field of vision. So that is what we call bitemporal loss. And that usually could happen from a pituitary mass, because remember, the pituitary is sitting right in that area. So if you want to simulate that, just basically put your hands in front of your eyes about halfway and see how much visual, and look straight forward and see how much your vision is affected. How much can you actually see without turning your head? So that's what that would kind of be like. And I'm sitting here doing these on myself like you guys can see me, but to tell you how to do it, I need to do it myself, so humor me. All right, if you have a deficit in your optic nerve coming from the optic tract, you end up with homonymous hemianopia. All right, and it's affecting the nerves on the right side, and if you notice, both of these are red. So that means one is coming from the left eye, taking into account the lateral visual field of the left eye and the medial visual field of the right eye. Okay, so what you'd have left is your lateral visual field or medial visual field of the left eye and lateral visual field of the right eye. So you would only see half on the same side for each eye. And I guess a way to simulate that is to put your hand over the left side of each eye and kind of see what you can see with that. It looks like you almost have like bars in your vision. All right, for the next two, I'm not even going to attempt to explain how to see that, because as you can see, it's getting a little more complicated. If we have visual defects in our optic radiations coming from the lateral geniculate nucleus, we end up with what we call quadrantinopia. This would happen on the right parietal side. All right, and this is what your vision would look like. You notice that the vision in this side is intact, but the vision on this side, half of it is gone. So you end up with this weird kind of almost pie-shaped vision. And the same thing if you have an optic radiation, a deficit in the optic radiation in the other nerve. You have a similar type thing, only where the vision is missing is in the superior part as opposed to the inferior part. And then finally, your IOs ask about being able to explain what a slit lamp is and how it works. I am not an optometrist. I've never used a slit lamp, and so I'm not going to try to insult your intelligence by trying to explain it myself. So what I'm going to do for this video and for the other two videos that I wasn't able to play for this lecture, I'm going to post them, in addition to being in the PowerPoint, I'm going to post them as links within the eye section of your ClinMed module for the eye, so that you'll be able to directly click on them and view them at your leisure.